Look at the physical mass of a growing forest canopy. Every single leaf, branch, and trunk you see is constructed primarily out of just two elements, carbon and nitrogen. But there is a biological catch. This diagram shows a plant leaf drowning in nitrogen gas. Our atmosphere is 78% nitrogen, yet in this gaseous form, those molecules bounce right off the plant. It cannot absorb a single drop. The same limitation applies to carbon. The forest floor is littered with carbon-rich organic matter, but roots don't have mouths. They cannot physically chew on dead wood or dry leaves as they pile up on the surface. Without an intermediary to process these elements, a forest would starve to death while completely buried in its own food. To find that intermediary, we have to look underground. 59% of the planet's entire biodiversity exists down here, completely out of sight. The sheer density of this microscopic workforce is difficult to conceptualize. Scoop up a single teaspoon of productive soil, and you are holding up to 3 billion individual bacteria. Take 1 gram of that same dirt, and it contains up to 700 kilometers of microscopic fungal threads packed tightly together. These dense microbial communities act as biological brokers. They possess the chemical tools to convert sequestered elements into usable forms, closing the distance between dead resources and living plants. This active, living barter system turns static dirt into the primary engine of terrestrial survival. Let's look at how this market actually operates, starting with the fungi. Fungi face a major biological deficit. They lack chlorophyll, meaning they are completely unable to photosynthesize their own food from sunlight. To survive, many fungi act as decomposers. They release enzymes into dead organic matter, breaking down tough plant cellulose to extract sugars. As they eat, they exhale carbon dioxide back into the atmosphere. The mushrooms you see emerging directly from the dead litter are just the fruiting bodies of this massive, invisible digestive network below. But this schematic shows a different strategy. These are mycorrhizal fungi. Instead of hunting dead matter, they solve their food deficit by physically invading the roots of a living host plant to set up a direct trade. The transaction works like this. The host plant uses sunlight to manufacture sugars internally and then pumps them downward, directly feeding the attached fungi. In return, those microscopic fungal threads scavenge the surrounding soil far beyond the plant's own reach. They absorb moisture and hard-to-find minerals, trading them back up into the host plant. As this expansive network explores the dirt, it secretes a sticky protein called glomalin. This protein binds loose soil particles together, holding the earth in place and preventing erosion. This localized fungal survival strategy acts as a massive global vault. It safely stores vast quantities of carbon deep within the structural architecture of the soil itself. While fungi are excellent miners, they cannot process the raw nitrogen gas trapped in soil air pockets. That specific bottleneck is handled entirely by soil bacteria. Step 1 is nitrogen fixation. Specific bacteria pull useless nitrogen gas right out of the soil pockets and convert it into organic nitrogen. To do this efficiently, they invade the root tissue of plants like legumes to set up colonies. Like the fungi, these bacteria perform this heavy chemical lifting as a direct trade for the plant's carbohydrate supply. Step 2 involves different specialized bacteria that scavenge dead matter. This flowchart outlines nitrification. Bacteria consume decaying organic proteins to manufacture their own energy. In the process, they convert them into ammonium and finally oxidize them into a chemical called a nitrate. Those resulting nitrates are the exact chemical form of nitrogen that higher plants require to grow. But if these bacteria kept manufacturing nitrates indefinitely, soil nitrogen levels would eventually become toxic. The system needs an exhaust valve. Step 3 is denitrification. Deep in oxygen-free pockets of the soil, anaerobic bacteria survive by eating those excess nitrates, converting them back into harmless nitrogen gas that vents safely into the atmosphere. These bacterial populations operate a relentless, invisible assembly line. They extract nitrogen, process it to fuel plant growth, and vent the excess to regulate global levels. Zoom out to the scale of the forest, and you can see how macro-level biogeochemical cycles are simply the aggregate result of trillions of these microscopic trades. Larger organisms scale up this efficiency. As an earthworm burrows, it physically mixes this microbial matrix. It ingests organic matter, stabilizes soil aggregates, and buffers the pH levels to keep the habitat neutral. Even burrowing vertebrates participate. By breaking up the packed surface, a gopher increases the soil's porosity. That allows rainwater and fresh air to easily infiltrate the ground, replenishing the microbes below. The carbon and nitrogen cycles are active, highly engineered biological processes driven entirely by organisms trading resources to survive. Every handful of Earth constitutes a dense, interconnected biological web. When that microbial balance is damaged by harsh chemicals or overtilling, the barter system breaks down. Plants are physically cut off from the nutrient cycles locked right beneath them. More than half of the life on Earth lives in the dirt, working tirelessly in the dark. Soil is a highly evolved living matrix. Without this microscopic workhorse,